just to give you guys a quick rundown, my lab specializes in the design and adaptation of high-throughput quantitative approaches to better understand host pathogen interactions and how those interactions lead to differences in disease severity and treatment outcome. So it's kind of like if a bug is going around the office and you have a head cold, Susan next to you, she's just living her best life, and Karen in the cubicle next to her is out for a week. Right now, we tend to think of these variations as bad luck or as a consequence of lifestyle, some sort of moral failure on behalf of those people. And then these variations, rarely do we ever think of in terms of the biology. Karen always gets sick is a fairly medieval way of thinking about infectious disease, but in truth, that's how oftentimes we think and talk about it. And these perceptions have dramatic impacts on the products we make, the research we do, and what our priorities are. So it's really important to understand this variation and what it means. So with that in mind, I just wanted to give you a quick rundown on what our lab is doing in regards to trying to understand the human components and how they relate to HIV infection. So HIV really needs very little introduction. There are currently over 37 million people living with the virus worldwide, with over 1.5 million new infections last year alone. The virus, if left untreated, infects and depletes CD4 positive T cells, causing acquired immunodeficiency syndrome or AIDS. So while there are a large number of highly effective antiretroviral therapies now, these therapies are not cures. So once someone is infected, they are infected for life and subject to the downstream physical, mental, economic, social, and legal consequences of that status. So that is not to in any way belittle the amazing progress that antiretroviral therapy has done. Really it has changed the face of an epidemic from what once was a death sentence to what is now a manageable condition. In fact, if somebody is on antiretroviral therapy and they're successfully suppressing virus replication, they virtually cannot transmit the virus. And the World Health Organization has recently adopted this as policy, wherein if the virus is undetectable in the blood, it is untransmittable. Undetectable equals untransmittable. And that's basically the principle behind the global effort to end the epidemic. The idea that if we can get enough antiretrovirals to enough people, we can stop the virus from spreading. And so it's been the goal of UN AIDS to achieve this 90-90-90 goal by today, by 2020. Essentially, to have 90% of the people infected to be diagnosed, 90% of the people diagnosed on treatment, and 90% of the people on treatment to achieve viral suppression. And sadly, we are nowhere near actually achieving those goals. Currently, it's estimated that about 70% of the people living with the virus are diagnosed, only 77% of which are on treatment, 82% of which have achieved suppression. And while these numbers sound pretty good, they result in really dramatic failures on our part to stop transmission of the virus. So our original goal in 2020 was to have less than 500,000 new infections each year. We're currently over 1 million new infections a year above that goal. So all of this is just kind of to say that as amazing as antiretroviral therapies are, they're just not enough. In the first place, anybody who's infected and on antiretroviral therapy has to take a pill every single day for the rest of their life, which is vastly too expensive and just completely infeasible in many resource-poor settings. Furthermore, even if somebody is on these kind of therapies, these ART drugs are not perfect. They do not restore normal immune function, nor do they completely fix or repress virus replication. And this leads to systemic inflammation and increased rates of neurodegeneration, liver and kidney failure, cancer, and a host of other comorbidities that together decrease lifespan. So this is all basically to say that to improve patient health and provide a lasting solution to the epidemic, we really need to invest in new curative and improved therapeutic solutions. And these interventions really need to account for the host as much as they account for the pathogen. And what I mean by that is we can no longer just think of this virus as sitting in a test tube and try to stop it. We really have to think of the virus as a two-part system. It's virus and host. That's the whole nature of infectious disease. So like all viruses, HIV relies on components of the host cell to replicate and survive. HIV relies on hundreds of different genes, proteins, and metabolites in a human cell to successfully replicate. It turns an infected cell essentially into a virus production factory, and at the same time is able to inhibit our immune defense with only 9 simple genes. How do we do this? How does the virus do it? And can we use some of these pathways to develop new therapeutics and new cures? This is something that biologists have been trying to figure out for decades, and that's actually the most difficult part. So we've been looking and trying to identify all of the different genes that HIV uses to infect a human cell, and we've been pretty poor at it. So just about a decade ago, so this was right in the sort of heart of genome-wide RNAi was the big thing on the scene. 4 big labs came out with 4 big papers, each looking genome-wide using these RNAi platforms to identify which genes HIV uses to replicate. They each identified in these high-profile papers roughly 300 to 400 genes that the virus required to replicate. When you take those data sets and overlap them, they all agree on exactly 0 genes. So what's the problem? This threw the field into a crazy tizzy because if these giant labs can do these giant experiments, genome-wide screens, and then agree on 0, that's a problem. So a bunch of post-mortem studies came out trying to identify exactly what went wrong. And basically, they came to 2 conclusions. One, they relied on RNAi. That's limited by transient activity, off-target effects, and incomplete knockdown. But then 2, these cell lines are not authentically recapitulating the intracellular environment of the host cell that HIV normally infects. So these are the cell lines they used in these studies, everything from HeLAs and HECs to Jurkat T cells. So just to emphasize this point, here are some EM images of a HeLa cell in the upper left, a Jurkat cell on the upper right, and then a primary T cell at 0, 24, and 48 hours post-stimulation. HIV can really only infect that cell, the activated T cell. It doesn't naturally infect HeLa cells or Jurkat cells, but oftentimes we rely on them as models in our research. And even looking at the pictures of them, you can see the gross morphological differences between these cells. And at the molecular level, it's the differences are even greater. So based on all of this, our lab teamed up with Nevin Krogan and Alex Marson at UCSF, as well as with Jennifer Dowden at UC Berkeley, to develop a high-throughput CRISPR-Cas9 platform for editing in primary human T cells. How this platform essentially works is we isolate T cells out of human blood. These T cells are then activated. In the meantime, we isolate Cas9 protein in vitro and synthesize guide RNA. These can be complexed together to form stable Cas9 ribonucleoproteins. These proteins that are delivered to these cells by electroporation to result in a polyclonal knockout pool. These can then be expanded. You can verify the knockout at the genome or the protein level, and then use them for downstream phenotypic assays such as HIV infection. So just to give you a quick rundown on the platform, cell number in a simple little 96-well cuvette, you can electroporate somewhere between 100,000 and 1 million cells, 20 microliter reaction volumes. Efficiencies, we're getting near complete ablation. You can do each one of these in less than a couple hours. You can do them 96 wells at a time. We're currently doing 384 at a time. Everything is commercially available. Right now, we're using the Amax Elanza platform, but it's adaptable to any electroporation device you might have. You can multiplex it, so you can do up to quadruple or quintuple knockouts if you wanted to. Toxicity is fairly low. You can freeze thaw these RNPs however much you like, and the cost is relatively minimal. So, some proof of principle. So how HIV enters a cell is it binds to CD4 as the main receptor. Then it binds to a co-receptor, either CXCR4 or CCR5, depending on the strain. So we decided to knock out CCR5, the co-receptor, just to show that this could indeed be done. So here we're looking at FACS plots on the left, staining for CCR5 on the y-axis and CD4 on the x. As you can see in our normal T cells, there's a subpopulation expressing CCR5. You treat with a control, such as Cas9 only, that's still there. You treat with our CCR5 guide RNAs, and expression is ablated. It does not impact the cell surface expression of other markers, such as CD4 or CD25, which is a marker of T cell activation. Now, if you look over then at infecting these cells, such as in the blue histogram, these are cells infected with the CCR5 tropic HIV-1 virus. And you can see in our control reaction, we have complete infectivity. And then in our CCR5 knockout cells, we see almost no replication. And this can be completely rescued simply by changing the envelope on the virus. So it's not that these cells are sick or dying. It's truly because of this specific gene edit. So this is great, and we can do this all one at a time. But we really wanted to know is, can we do this hundreds of genes at a time? So we decided to go ahead and knock out roughly 430 HIV human protein-protein interactions. So this is just a network map of the physical interactions HIV is thought to make in a human cell upon infection. There are over 400 different genes that the virus is supposed to be able to physically bind to, but nobody has ever actually tested to see if these were genetically required. So we decided to knock these all out because they should be functionally important. We would expect them to have an effect. This provides a mechanistic handle for functional follow-up because we have physical and functional genetic data. But then also we wanted to just kind of ask the question, as mass spec becomes more and more used, how many of these hits are actually going to prove to be translatable? So this is what we decided to do. This was basically our experimental design. We had 430 genes that we were able to design guides to, 3 CRISPR RNA per gene plus controls. Those were synthesized in 96 well arrays, electroporated into activated T cells from at least 2 to 5 donors per well. These were then activated and expanded, replica-plated, and then infected with a GFP reporter virus, which was monitored by automated flow cytometry over time. At the same time, we collected genomic DNA from these cells, and we used deep sequencing to calculate our efficiency of knockout. So just briefly regarding the efficiency, how we did this was we have bioinformatically designed primers flanking the target gene site. We amplify across, add index primers, spray purify, do deep sequencing, and we end up with a lot of reads kind of like is shown in that cartoon on the right. So CRISPR-Cas9 relies on the endogenous repair machinery to repair those edits. But based on the reads, you can calculate your percent insertion deletion and your percent efficiency of your guide cutting. And we were able to get reads for about 83% of our knockouts. So you take all that data, look at it, and right here we're showing the most efficient guide per gene in pink in the histogram. So on average, we were able to get a knockout efficiency of at least 75% in our primary T cells. And if you look at the variation donor to donor, as shown in the dot plot over on the right, you can see that we have very high correlation moving from one patient to another. You do see a few points where one donor is way better at editing than the other. When you actually look at the sequencing data, that's usually because there's a SNP in the CRISPR binding site in one patient, but not the other person. So that's all well and good. We have our efficiency data. Now let's take a quick look at our infectivity. So like I said, we incorporated the same 6 controls on every single plate that we did. 3 non-targeting, which are shown on the left. We get very tight reproducible rates of infection. And then 3 control genes, which we know dramatically decrease HIV infectivity. So they all look good days 3, 5, and 7. Looking at the results from every single guide, we identified many guides that decrease infection, a few that increase infection. They stay relatively stable over time, as could be expected, not infected. But then the cool thing is you can take your efficiency data and plot it on the x-axis relative to your infectivity data. So now the more efficient a guide RNA is, we're seeing more and more perturbations to the infectivity of these cells. So based on a number of titration experiments that I can't go into right now, we were able to, identify that we need 30% editing in order to see a phenotype in our assay. So assigning a 1.5% FDR, we can identify our true hits, which are shown here in blue. So if you take these guide RNAs then and map them back to the gene level, we identify 86 genes in this network map that are essential for HIV replication, or roughly 20% of our HIV protein-protein interaction map. So you can take that functional data and now put it onto the physical map, and you have a functional map of how HIV is replicating directly in a primary T cell. And of the 86 host factors identified, roughly half had been validated in previous mechanistic studies, but the other half had not since been reported. And we've been slowly working through our way and publishing on the mechanism of these, but we have been able to recapitulate a lot of really well-known biology. So for example, the HIV transcription factor TAT binds to the PTEF-beta complex to stimulate transcriptional elongation. And of course, when we knock out members of PTEF-beta, we see decreases in infection. We knock out negative regulators of that, as shown in red, we increase infection. So all very consistent. One really surprising thing that we found, and one of the last things I wanted to note, is we do see pretty dramatic donor-to-donor variability in specific host factors, meaning that this virus is using different genes in different people to get the job done. So for example, when we look at this gene FBX06, here we can see in 4 different donors, A, C, D, and E, no difference between the blue and black line, meaning no difference between the non-targeting control and knockout. But in donor B, for whatever reason, this gene is incredibly important for this virus to get by. And we have absolutely no understanding of this as of yet, but it's interesting and important to consider when we're beginning to develop therapeutics that would target any of these pathways. So moving forward, what we're hoping to do with this is now we're taking sequencing data from these patients and mapping single nucleotide variants onto these important complexes and genes and beginning to knock in single nucleotides one at a time to identify how these variations in the human genome can contribute to differences in disease outcome. So with that, I just want to leave you with this thought. High throughput gene editing in primary human cells is readily doable. If your company is not doing this to support their biological pathways, identify their drug targets, et cetera, they really should be, because primary cells are very different than your cell line models. With that, just wanted to thank people in my lab who contributed, all our collaborators and funders. Thank you much.